Hello, everybody. I'm going to focus my talk specifically on measurement of iron in the liver. I'm going to talk about a new technology that is now available, MRI-based technology for measuring iron load, that I think overcomes at least some of the challenges that we've faced recently. I first of all want to declare that I have a competing interest. I am employed part-time by Resonance Health Analysis Services, the company that provides FerriScan. So this is an overview of what I'm going to talk about. I'm going to start off by asking the question, is it difficult to measure liver iron concentration with MRI reliably? And if it is difficult, what are the current pitfalls that we're facing that make it difficult? I will then present some potential solutions that address some of those pitfalls, and they include the development of an MRI image data acquisition quality assurance software module. So this is something that checks the quality of the data that's being collected on the MRI scanner. And I'll also talk about the training and testing of an artificial intelligence convolutional neural network that automatically analyzes MRI images. Now that last bit's a bit of a mouthful, but you probably heard about artificial intelligence. We use it a lot in our everyday lives now. Anybody who's used Siri on their iPhone or something like that is using artificial intelligence. And then we'll look at how well these potential solutions work. And we'll look at a particular study that tests this new technology called FerriSmart against FerriScan as the reference standard in a multi-scanner study. And I'll quickly talk about the regulatory status of this new technology. So let's start off with the question, is it difficult to measure liver iron concentration with MRI reliably? The answer probably depends on who you ask, but I think there's a lot of evidence to suggest that for many centers, it is difficult. It's not an easy task, and I'll show you why. First of all, let's acknowledge that there have been many research papers published that talk about methods for measuring liver iron concentration with MRI. But with few exceptions, those papers describe a method that has been developed on a single MRI scanner. And that does introduce some problems. All of these methods comprise, first of all, a method for acquiring the image data on the scanner, and then a method for analyzing those images to calculate an MRI parameter such as liver T2 star or R2 star or T2 or R2. We don't right now need to exactly understand what those numbers are. Suffice it to say, it's something you can calculate from these images. And the published data indicates that in the hands of experts, these methods usually give useful results. In other words, that parameter, that MRI parameter, be it T2 star or R2 star, correlates quite strongly with the liver iron concentration. Now, what I'm showing in this graph are the results from 22 different published studies that show a correlation between liver T2 star and liver iron concentration. That liver iron concentration might be measured by biopsy or by FerriScan. And what we can see on this graph are all the different calibration equations that have been derived from those studies. So these lines, each line is from each of the 22 studies. It shows how the liver iron concentration relates to the T2 star value. And what you can see is that these relationships are very different in the different studies. And the reason for that is because both the method that the researchers have used to acquire the images and the method that they've used to analyze the images result in a different dependency of T2 star on the liver iron concentration. So the important point here to take away is that the MRI parameters like T2 star are dependent not only on the iron concentration in the tissue, but also on the precise details of the settings used on the MRI scanner to acquire the data. And this picture that I have at the top is a typical example of what an MRI scanner control console looks like when the radiographer is acquiring the data. You can see there's all sorts of different settings they have to adjust, and each one of those needs to be correct or match the technique that's used to generate the calibration. And similarly, the T2 star is dependent on the precise details of the method used to analyze the image data. And there are typically an equal number of variables that you can use to analyze the data. So what are the current pitfalls? The pitfalls are that when using a calibration curve from the literature, which is what most people do, because it's quite time consuming to generate your own calibration curve. But if you're going to use a calibration curve from the literature, then you must make sure that both the image data acquisition and the image data analysis methods match those used in the study that generated the calibration curve. That sounds simple enough, but as we can see from the second dot point, sometimes the scanner hardware that a radiologist has may not be compatible with the given method that was used to give the calibration. So for example, there's one particular parameter that's very critical. It's called the first echo time. Some scanners cannot achieve the first echo times that were published with those calibration curves. Secondly, the image data analysis software that the radiologist has may not have all the required attributes to replicate the type of analysis that was used in the calibration study. And we should remember finally that there are multiple different possible variations of scanner settings for the data acquisition and multiple possible variations in the way we analyze the image data. So you put all of that together, and it's actually quite a complicated, task. So this then results in the possibility of extreme errors. And this is a study that was carried out by Trang and colleagues in 2017. And what they did here was they compared a T2 star technique for measuring liver iron concentration with the FerriScan technique on two different scanners. One was a Philips scanners, those are the closed circles, and another scanner was the Siemens scanner, the open circles. And what we can see is on the vertical axis, the liver iron concentration measured by the T2 star method, and on the horizontal axis, the liver iron concentration measured by FerriScan. And the black line is what we call the line of equivalence. And we expect, in the best case scenario, the data points to be scattered around that black line. But in the case of the Philips scanner, for example, we see that down at the low levels, there's a little bit of correspondence between the two measurements. But as we go to high liver iron concentrations, we get extremely severe errors. So for example, let's take this patient here. With FerriScan, the liver iron concentration was extremely high at 50 milligrams of iron per gram dry tissue, whereas the T2 star method gave a value of around about 3 or 4 milligrams of iron per gram dry tissue. In another study from Sri Lanka by Padania and colleagues, they again compared a T2 star method for measuring liver iron concentration with FerriScan. And if we look at these patients up here, the T2 star method's giving results of around about 3 milligrams of iron per gram dry tissue, whereas the FerriScan is giving results of about 40. So very extreme errors. And in this case, a really unusual relationship, a negative relationship between the two measures. So I think these data show that you can get serious problems with liver iron concentration measurements. So I now want to present a potential solution to some of these pitfalls. Just remembering that, looking at this picture again, this is the control console at a particular MRI scanner. We want to make sure that all of the settings on the scanner are correct for the particular method of liver iron concentration measurement that we're using. So what we have done is we've developed a software package that uses artificial intelligence, and it's fully automated. So this system, the commercial name is FerriSmart. The technical name is DLA R2-MRI. And the analysis is fully automated in that the radiologist collects the data and then literally drags and drops onto the software, and a report is generated. The technique of data acquisition is in fact identical to that for FerriScan. And that's important because the FerriScan method has been shown to be achievable even on quite old 1.5 Tesla scanners. So you don't need a modern up-to-date scanner to use it. What the software does, first of all, is that it checks that the image data have been acquired correctly. And if the data have not been acquired correctly, the software gives immediate feedback to the radiographers or radiologists and tells them what errors have been made. So it gives the radiographer an opportunity to correct that mistake on the spot. Importantly, a liver iron concentration result is not given if the data have been acquired incorrectly. If the data have been acquired correctly, then a liver iron concentration report is generated within seconds of uploading that correct data. Importantly, this technology has regulatory approval from the FDA in the United States and CE mark in Europe. So how well does it perform? Well, we carried out a study on 1,395 eligible consecutive MRI data sets that were submitted to Resonance Health for routine clinical analysis by FerriScan. Those data sets were from 63 different MRI scanners, and none of those scanners were used in the training of the artificial intelligence system. The etiologies for iron overload that were reported by the radiologists included the thalassemias, hereditary hemochromatosis, sickle cell disease, myelodysplastic syndrome, and other conditions or unknown conditions. And then we also tested the repeatability of the device by assessing 60 participants. That was 50 patients with iron overload etiologies and 10 healthy controls. And each of them was measured twice. And then we compared to the results to see how repeatable the data were. So first of all, the quality control module. What we can see here in this table is that 1,281 cases, in other words, 91.8% of those of the cases studied passed both the FerriScan and the FerriSmart quality control processes. 1.6% failed both. So that's where both were in agreement with the failure of the quality of the data. 5.3% passed FerriScan but failed FerriSmart. Now, this looks like it could be a problem, but it turns out that many of those cases are rather trivial mistakes. For example, the radiologist uploaded 2 patient data sets instead of 1. So this is an easily correctable mistake. And then finally, 1.3% failed the FerriScan but passed the FerriSmart. This is perhaps the cases that we should be most concerned about, but fortunately it's a rather low percentage. And this is then what the correlation looks like between the FerriSmart data on the vertical axis and the FerriScan data on the horizontal axis. The solid line is the line of equivalence. And you can see that there are no, or not a large number of catastrophic failures, and the sensitivity and specificity for detecting liver iron concentrations above these clinically relevant thresholds of 3, 5, 7, and 15 milligrams of iron per gram dry tissue are all generally in the 90s, apart from the sensitivity for the 15 milligram per gram dry tissue threshold. There is a slight bias between the 2 techniques. The FerriSmart reads on average about 93% of the FerriScan data. And this is what the repeatability data look like. So there's a reasonably good repeatability between the 2 techniques. In summary, what's important about this is that the repeatability is much better than that of biopsy. Currently, the regulatory status for FerriSmart is that it's available, it's been approved by the FDA in USA, and it has CE mark in Europe. It is currently marketed by Resonance Health Analysis Services, and they are trying to aim the pricing, especially for low-income countries, by keeping its price below 10% of that of the FerriScan price. And Resonance Health is working with TIFF to identify sites currently that wish to try the technology free of charge for up to 12 months. So in summary, the repeatability of this new technology is significantly better than that of biopsy. While there is a bias between the new technology and FerriScan, it doesn't result in unacceptable sensitivities and specificities. But the bias does mean that the 2 techniques should not be used interchangeably. And finally, I would mention that it is available for clinical use now. So we're keen to see people try it out. And with that, I will end my presentation and thank you for your attention.